First, I'd like to thank Dr. Aggarwala for his excellent presentation on metastatic melanoma. I'm Alain, Rolland. I'm Executive VP, Product Development at Vical, and I'm going to show you how we have developed a novel immunotherapeutic for melanoma. Allovectin is Vical's lead product in a phase 3 pivotal trial. It is a first-in-class immunotherapeutic currently evaluated in metastatic melanoma patients. Allovectin is a plasmid-based immunotherapeutic that expresses two genes which together form an MHC class I complex. It is formulated with a proprietary lipid-based system that facilitates the delivery of DNA into the target tumor cells. It has a convenient presentation as a single vial, stored refrigerated, and we are using a conventional needle and syringe administration into a lesion. The initial development of Allovectin that I will review today is focused on metastatic melanoma, but as an immunotherapeutic, it could have potential applications in other accessible, solid, and immunoreactive tumors as well. Allovectin is injected into a single lesion to produce both a local but also a systemic effect through 6 weekly injection cycles. The product is used in a physician's office, as we heard from Vijay, in an outpatient setting, and it does not require any pre-medication or post-medication, such as antiemetics or hydration. One of Allovectin's hallmark has been the remarkable safety profile to date, observed in multiple trials with multiple doses as well. Allovectin has a unique set of mechanisms of action. Really what it does, it teaches the immune system to recognize and destroy the tumor cells. First, the HLA-B7 flags the tumor cells as foreign in a way very similar to the transplant mismatch. This leads to an immune reaction against these tumor cells. Then, beta-2 microglobulin also restores the effective presentation of the tumor-associated antigens from the patient, making these tumor cells more visible to the immune system. And finally, the lipid DNA complex that we are using induces a danger signal that further boosts the immune system against these tumor cells. So in other words, those different actions result in a local and a systemic recognition of the tumor cells by these educated immune cells. Allovectin mechanism of action is complementary and possibly synergistic to other anti-cancer therapies. We are very excited, as we heard, about the recent success of Provenge, for instance, and ipilimumab, but combination modalities, as Dr. Aggarwala pointed out, will likely remain the standard of care in oncology. So in the example shown on this slide, the T-cell activation generated by the anti-CTLA-4 antibodies, such as ipilimumab, could work in concert with Allovectin 7 by boosting the tumor cell destruction by these T cells that have been activated. The safety and the efficacy of Allovectin at high dose, the 2 milligram dose, were evaluated in a phase 2 clinical trial, a very large phase 2 with 127 patients. Most of the previous trials that we have done at Vical were conducted with a much lower dose, 10 microgram dose, which is about 200 times lower than the dose that we are using or we have used in phase 2. The efficacy was evaluated in 127 stage 3 and stage 4 metastatic melanoma patients, and several response endpoints were measured by strict RECIST criteria. Overall survival was also assessed in this phase 2 clinical trial. Allovectin was very well tolerated, as you can see on this slide, when compared to other treatments such as DTIC and ipilimumab. The 11.8% response rate achieved with Allovectin 7 compares very favorably with historical data with chemotherapies, for instance, such as DTIC, or immunotherapies such as ipilimumab. All the responders to Allovectin had a durable response for at least 6 months. As presented by Dr. Aggarwala and as shown on this slide, chemotherapy results in a very short-lived response. For a subset of chemo-naive patients that we are targeting in phase 3, the durable response rate was about 17%, so that's highly remarkable. Importantly, in this large phase 2 clinical trial, the median survival was 18.8 months, which is remarkable when you compare to historical data in metastatic melanoma population, where the survival range is typically from 6 to 11 months. In fact, this Kaplan-Meier graph presents the very promising median survival of 18.8 months obtained with Allovectin 7. Even though 63% of our patients receive 1 cycle or less of Allovectin due to progressive disease, based on the strict RECIST criteria, at least half of the patients showed a substantial survival benefit compared to historical controls. The responders in particular had prolonged survival ranging from 18 months to more than 7 years, and the median survival, as you can see on this graph, was not even reached for the responders. We further compared Allovectin phase 2 survival data to a recently published analysis of 42 phase 2 clinical trials in about 2,000 metastatic melanoma patients. In this publication, as shown on this graph, the mean 1-year survival was 25%. No statistical difference was observed with any of these treatments, since all these 1-year survival points, all these blue dots, basically fall within the 95% confidence interval. In contrast, if you look at the Allovectin 7 1-year survival of 65%, this is well above the range, showing a statistically significant improvement in comparison to this historical data. Even the 2-year survival for Allovectin, which is 43%, is still better than the 1-year survival for all of these published data. So what did we learn from these phase 2 results in order to maximize the phase 3 design for the first-line indication? First, we selected the healthiest metastatic melanoma patient. We heard normal LDH is important. We selected normal LDH. No brain mets, no visceral mets, no liver mets. And we also selected chemo-naive patients with more intact immune system. And this to keep the patient on study as long as possible in order to maximize the chance of benefit from the immunotherapy Allovectin 7. We injected a single lesion with a full dose, 2 milligrams, to optimize the anti-tumor effect. We also modified the RECIST criteria to help keep the patient on study for at least 2 cycles. 2 cycles was the median time to response in the phase 2 study. We also allowed resection of a stable lesion after 32 weeks to verify if the lesion is really a lesion or whether it is only scar tissue. And then we used, as we heard from Dr. Agarwala, a primary endpoint that really favors durability of response observed for immunotherapies. So the phase 3 pivotal trial design was accepted by the FDA under a special protocol assessment and SPA. It is intended to show superiority of Allovectin versus first-line chemotherapy, DTIC or temozolomide. The trial is powered at 90% at least to show a benefit not only in response rate but also in survival benefit. The trial over-enrolled 390 patients by February of this year due to a large number of subjects that were in screen both in the US and in Europe. And Angis, our partner in Japan, has fully funded the clinical cost of this phase 3 clinical trial. We expect, as Vijay mentioned, to lock the database in the second half of 2011, and shortly thereafter we will announce the p-value. So in summary, Allovectin is the first-in-class immunotherapeutic with a unique mechanism of action that is complementary to other anti-cancer therapies. Allovectin is used in an outpatient setting and it has a very safe profile. We have focused initially on metastatic melanoma, but Allovectin has potential indications in other solid tumors. We have obtained very promising results in this phase 2 clinical trial, and the pivotal phase 3 trial has completed the enrollment early this year. We have obtained also orphan drug designation and fast track designation in the US, and we are now getting ready for commercialization of Allovectin 7. So I'd like to thank you.